So once again, as we will move on to the next session, as the previous chair has already introduced Dr. Rosita, I will proceed to invite her once again to now chair the next session. Dr. Rosita, the floor is all yours.
Yeah, okay. Yeah, thank you very much, Dorothy. Yeah. First of all, I would like to say thank you to the organizer for this virtual workshop on the management of bacterial disease in banana 2022 and for the brief introduction. It is a great honor to be chairing this session. Yeah, I'm Dr. Rosita, as they mentioned earlier. We are very delighted to have three prominent speakers from China, Philippines, and Indonesia. Before we proceed, may I reinstate again to our respectful presenter to present in allocated time frame provided, 15 minutes with 5-minute Q&A. Yeah? Our first presenter is Dr. Yunhao. Hopefully I pronounce it in a very good Chinese. Yeah, Yunhao from Innovative Institute for Plant Health, Zhongkai University of Agriculture and Engineering, Guangdong, China. Yeah. So before that, may I briefly introduce our first speaker, Dr. Yunhao completed his Master Science and PhD in Sun Yat-sen University in China. In research, he interested in understanding alteration plant signaling, metabolism, hormonal balance, and proteomic changes underlying both immune response and bacterial virulence. Again, activities which generate fundamental knowledge and to deploy applied strategies to achieve sustainable solution to fight plant disease. For his research, he was awarded with several numbers of national fundings from China and published several numbers of impactful papers. Yeah. Without any delay, may I call Dr. Yunhao to present his slide presentation entitled Pathogens Profile of Klebsiella variicola, the causative agents of banana shaped rot. Please welcome Dr. Yunhao. The floor is yours.
Thank you for Dr. Rosita for the nice introduction.
Yeah.
Okay, good afternoon or maybe good morning for everyone. It's my great honor to have this opportunity to make this presentation here. Yeah. And in the next 60 minutes, I will introduce one of our recent work briefly. Okay. Can you see the PowerPoint, right? Okay, we know the industry has experienced dramatic annual losses due to various diseases and design work of banana. Banana number one is one of the major banana categories in China and shows high resistance to the Fusarium root, so it's regarded as the ideal culture for growth in the problematic areas. However, in recent years, we noticed the emerging of one disease of banana in multiple regions of the world, and in our recent research, we found a new disease outbreak in the world, and we found that almost all the banana varieties planted in the southern China, including banana number one, has been suffering from a new disease that causes serious losses, and moreover, the epidemic area and the severity of the disease are increasing. So the typical symptoms of this disease are yellow-brown leaves, soft dark brown petiole tissue, and complete leaf collapse and plant death. And these symptoms can occur year-round, but more serious in the rainy season. We named this disease as banana sheath rot. Okay, in 2019 to 2022, we collected more than 30 samples of the diseased petiole tissues of the banana plants at different locations in the Guangdong and Guangxi provinces, where are the main producing areas in China. And 12 of these samples were showed in here, named as KV1 to KV12. And the pathogens were isolated from the symptomatic tissues, and seems that we isolate the same bacteria from these different samples, which form the white colony, circular, and smooth colonies on the plants, and we named these strains as the KV1 to KV12 according to the corresponding names of the samples. So to explore the species of this acidic strain, the molecular identification was performed using the 30 rDNA primers, and we noticed that most of all the 12 acid strains are most closely related to the Klebsiella variicola. From now, I may use KV short for Klebsiella variicola. The previous study has shown that the KV strains can fix nitrogen, and to test whether this is the case for our isolated strains, the nitrogen fixability of these strains was tested by the planting the bacteria on the Ashby's nitrogen-free agar medium, and we can see all these bacteria can grow, and the control bacteria, one of the Bacillus subtilis, was not, so indicated that these strains can fix nitrogen. And moreover, most of these strains can utilize a list of nutrients that is very similar with the typical strain in the genus of the Klebsiella. So based on the homological, molecular, physiological, and biochemistry analysis, the acid strain was confirmed to be the Klebsiella variicola. So to further verify the pathogenicity of this acid strain, we performed a punch inoculation of the leaves of two cardboards. That's short for fungi number 1, and the B is short for Brazil. At 40 days of the post-infection, we can see that the tissues around punch holes of the control plants remain healthy, whereas the chlorotic and necrotic lesions were present on the leaves, which infected by the acid strains. So at the 4 months of the infection, the typical symptoms observed during the field production were detected, and by isolating and identifying the pathogen at the disease site, we further confirmed that the pathogen were Klebsiella variicola. So how was the Klebsiella variicola? In 2004, KV strain was first proposed as a new Klebsiella species, and it's a gram-negative normal tail bacillus that can fix the nitrogen, and also regarded as a dominant bacterium in the fungus gardens of the leaf-cut ants. And also in recent years, they have been regarded as an optimistic pathogens which have been reported in the infections of the animals and the humans. And noteworthy that in recent, exactly the several past years, the Klebsiella is also shows the ability to causes the disease of various plant diseases, such as the bacterial soft rot, the plant soft rot, and the rhizomes rot of the banana. However, the most KV strains report data is found in the environment and the clinical samples, and there are very few case reports of the KV strains infections in the plants, and also the mechanism by which KV strains caused plant disease remain unclear. So to obtain more information about the strains, we sequenced all the genomes of these 12 acid strains, and we found that all the 12 acid strains belong to different FTRIs. So it's weird. Then we constructed the phylogenetic tree of the 12 acid strain, as shown in the right lines, along with the 440 other KV acids which are available at the NCBI genome database. According to the core genome alignment, we found that they are dispersed in the different branches of the phylogenetic tree, which pointing to the dissimilarity of these isolates. And noteworthy is that the evolutionary of the relationship of the most of these acid strains is also showed no consistency in the accessory gene content with each other or with the other acids from different plant sources. So This is not indicate that the kiwi strains might an opportunistic pathogen with the plant pathogenicity, but also remind us the kiwi strains may also a kingdom crossing pathogen, as the strains cause plant disease may also infect humans and animals. So to further confirm this notion, we generated an estimated pine genome analysis of the 441 analyzed isolates. Okay, we found that also the 12 isolate strains showed in the right lines have no consistency with each other. So then we investigated whether these isolate strains share a common absence of the virulence factors or the effectors. Also, briefly, we found more than 100 potential virulence factors and more than 2,000 potential secrete proteins were in the pine genome of the isolates, and some of them are carried by the 12 isolate strains as core genes. So we are working on to characterize the biological function or biochemical function of some of these proteins, which may help us to find out the mechanisms of the pathogenicity of these pathogens. In the banana plantations at the beginning of this disease, we found that a portion of the plant of the same variety in the same cultivated land will be diseased, while others will not. And in addition, as mentioned above, the 12 isolate strains showed different sequence types with each other, also implies that this disease may not have an obvious common origin or the chain of disease transmission. So considering that the isolate kiwi strains might also be a endophytic bacteria of the plant. So we guess and speculate whether the changes of other microorganisms would become a critical factor for the disease occurrence. So we sequenced and analyzed the microbial communities in the healthy tissues of the disease plant and the corresponding tissues of the healthy plant. For the two cultivars, Brazil and number 1. Firstly, we estimated the alpha diversity of the microbiome, and no significant difference was observed. And then we estimated the relative abundance of the genus Klebsiella, and also there show no significant difference between the different samples. However, we found that there are several other genus showed significant differential abundance between the healthy tissues of the disease plant and the corresponding tissues of the healthy plant. And noteworthy is that the increasing abundance in the general Herbacterium and the decreasing abundance in the genus Bacterium were detected in the healthy tissues of the disease plant in both cultivars. So then we constructed the microbiome interaction network at the genus level of the two cultivars. In Brazil, we found that the Herbacterium shows strong mutual exclusion with the most high abundance genera, and the Bacterium positively interacted with the most high abundance genera and indirect negatively interacts with the Klebsiella. And in the number 1, the Bacterium is directly excluded by Klebsiella, and the Herbacterium is indirectly mutual exclusion with Bacterium. So this result indicates that the Bacterium negatively interacts with the Klebsiella, and the Herbacterium might indirectly inhibit the Bacterium growth to promote the Klebsiella growth. So in recent years, the Herbacterium is reported as a plant endophytic genus that can promote the growth in the sugarcane, in rice, in maize, and in red clover. And it can also fix nitrogen and might affect the nutrient uptake of the host plant. So there are many similarities between the Herbacterium and Klebsiella. We speculate that there are also the co-relationships in the pathogenicity of the host plant. And taken together, this result indicates that and suggests that the banana sheath rot is caused by the imbalance of plant and endophytes and an optimistic pathogenic bacterium, which may provide the basis for the research and the control of this disease in future. Okay, now we want to reduce the occurrence of the misjudgment of this disease in the plantations and also reduce the possibility of the banana plants to carrying the bacteria in the stage of the seed reproduction and in the breeding. So we are working on to developing one system for rapid detection of disease agents based on the lab. And just mentioned about the genetic sequencing and the comparative analysis. We targeted and focused on several specific genes of the pathogenic kiwi strains and achieved several success under the laboratory conditions. And next, we will conduct the targeted verification in the plantations. And the last part, how about the research on the treatment of this disease? Also, under the laboratory conditions, we sequenced dozens of the bactericide for kiwi strains, and among them, we found that the acylacin, which is a broad spectrum plant biomimetic bactericide, also has the best bactericidal against the kiwi strains. And also, to our delight, that banana anaphytic Bacillus subtilis strain R31, which we used for the biological control of the banana Fusarium wilt, also has a direct antagonistic effect on the kiwi. So based on this result, we are now conducting the disease treatment experiments in the affected banana plantations with the combined medication.
Doctor, you have 1 more minute. Yeah.
Okay.
All right.
Yes, no problem. Our research team is in the Innovative Institute for Plant Health, Zhongkai University of Agriculture and Engineering. And we will think and hope there will be more research experiments, exchange and cooperation with you in the future. And okay, friends, thank each and every of you for your attention. Thanks.
Okay. So, all right. Okay. Thank you, Dr. Yunhao for the remarkable presentation. Yeah. Now we understand the science fundamental of Klebsiella variicola and might be in future to develop the management. Yeah. Now I open for Q&A for audience. Since the time is very limited, I only allow one question. Yeah. Maybe we can find out the question from the chat box here.
Madam Chair, I have my hands up. Can I ask a question?
Yeah, sure, Dr. Mr. Yeko. Please.
Okay, just Dr. Yunhao, thank you very much for the presentation. A little heavy for me, but it's okay. I just want, I couldn't hear you well just now, but I just wanted to confirm you were talking about the interaction with the microbiome and Klebsiella, yeah? The interaction between this bacteria and the other microbial stuff in the soil. So I can't hear you clearly. How do they interact? Like so much of certain microbiome will have an effect on its pathogenicity, or how does it work? The soil microbiome and Klebsiella itself. Yeah. Thank you.
Yes. Thank you. It's a good question. It's a key point for this research. Is really a key point for this research. During this research, we found that there are many other genera except the Klebsiella was increasing or decreasing in healthy tissues of the plant or the disease plant. And we found that the Herpesspirillum, the genera of Herpesspirillum, is increased in the disease plant at the early stage of the disease. And how the interacts work is we are now working on it, and we're trying to collect more the strains of the Herpesspirillum, not only the Klebsiella, from the disease samples from tissues, but it's not easy. We can isolate the Klebsiella variicola from almost every samples from we collected. There are just 1 or 2 from the 10 samples maybe can get the positive result of the Klebsiella. We have no idea why. Maybe the medium we choose best medium will be maybe the stage of the disease, maybe at the early stage of the disease. Klebsiella is very high abundance of the general. Maybe later stage it will be Klebsiella. So we are working on it. Yeah.
Okay, thank you. Thank you.
Yeah. Okay. I hope that I will answer the question. Yeah. Okay. Thank you, Doctor, for your excellent presentation.